The next topic is the role of telomeres and telomerase in aging and cancer by Dr. Shea. Dr. Shea is the Associate Director of the Harold C. Simmons Comprehensive Cancer Center at the University of Texas Southwest Medical Center in Dallas and Program Director of the Cancer Biology Graduate Program. He is also the Vice Chairman of the Department of Cell Biology, a distinguished teaching professor, and holds the Southland Corporation Distinguished Chair in Aging Research. Throughout his career, Dr. Shea has been interested in the relationship between aging and cancer, and in 1995 discovered that telomerase was present in the vast majority of human cancers. He then demonstrated in 1998 that introduction of telomerase into normal telomerase-silent human cells was sufficient for human cell immortalization without an increase of cancer. In 1995, he and his long-term collaborator, Dr. Woody Wright, received the Allied Signal Award for research on aging. Dr. Shea is a scientific advisor to LifeLength, a Madrid-based company founded in 2010 with the objective of commercializing the world's most precise telomere analysis technology. LifeLength is the only company in the world that offers a scalable technology to measure the percentage of critically short telomeres that reflect human biological aging, not chronological age. It's my pleasure to introduce Dr. Shea.
Well, thank you. As you can see, I'm not Mike Ditka, and it's a bit of a challenge to come before an audience expecting to see a very famous football coach, a newscaster, and just a very inspirational individual. And so you might be asking yourself, why would they ask this professor from Texas to come here and substitute for Mr. Ditka? And the answer is he and I go way back. We've actually have been long-term friends. Before he got into football, he actually was in a different league that he and I shared together, and I just thought I would show you that. We were both teammates on the Gladiators. That's my humor. So anyway, hopefully what I'll be able to do in the next 30 minutes or so is to entertain you in a different way by discussing the role of telomeres and telomerase and its relationship to aging and cancer. So by way of introduction, I do have a disclosure that I am a scientific advisor to LifeLength, a Madrid-based company involved in telomere length testing as a biomarker of biological versus chronological aging. So with that behind me, I just want to remind everybody, I think it's very obvious that today, as opposed to maybe 50 or 100 years ago, when we didn't have a protected environment, when most people, in fact, died of different types of infectious diseases and things such as that because we didn't have antibiotics, we didn't have good sanitation, we didn't have all of the wonderful biomedical interventions. So now that people are living longer, the question is, what really does limit the maximum lifetime of individuals? And what I hope to share with you today is some of our basic science and some of our clinical experience trying to work on this one field involving the ends of our chromosomes called telomeres. Now, this actually is a field that's been around for a long time. It was actually in the 1930s and 40s that both Dr. Muller and Dr. McClintock both received Nobel Prizes for basically what I show at the bottom of this slide, about describing the ends of all of our chromosomes. So all of our genes are on chromosomes. We have 46 chromosomes. Each end is different, and there are these repetitive elements at the ends of all these chromosomes. They don't code for genes. And these two scientists that each got individual Nobel Prizes were critical in showing that there were special complexes at the end of the eukaryotic chromosome. It turns out that these structures were termed by Dr. Muller telomeres. And what's interesting is all vertebrates have this same sequence at the end of all of our chromosomes. There's this sequence TTAGGG, and these are monotonously repeated thousands of times. And another Nobel laureate by the name of Jim Watson, who discovered DNA, was the first to show that the phenomena was called the end replication problem, that every time a human cell divides, your telomeres get a little bit shorter. And this is due to the mechanism by which DNA replicates. And there's other things. There are environmental aspects like oxidative damage and other in-processing events that can also lead to increased losses at the ends of our chromosomes. And the hypothesis that came out about 20 years ago is that senescence, or aging, occurs when some of these telomeres get very short. So what is the data? So how do you think about telomeres? Let's just give an analogy here. So everybody knows what the little plastic tips on the ends of shoelaces are called, right? Auglets. And they're covered these little plastic things. So you can think of your telomeres in all of your cells in your body as having, when you're young, good protected plastic tips. Now what happens as you get old, a subset of those plastic tips become unraveled. And when a tip becomes unraveled, or a chromosome becomes unprotected, that leads to a growth arrest of that cell, and that cell then can no longer divide and sustain and replace the tissues in the body. So the data actually evolved almost 60 years ago with Leonard Hayflick, who was at the Wistar Institute in Philadelphia. And Dr. Hayflick overcame a long-standing dogma that cells extracted from the human body were in fact immortal. That was the thought collective in the early 1950s. His experiments clearly showed, it is illustrated in this slide, that in fact normal human cells taken from the human body actually have a limited capacity to divide, and that when the cells reached a certain number of doublings or passages, the cells would look like here on the right. They would get big, and they would actually secrete a whole different constellation of proteins that's quite different from what you would see in a young quiescent cell. And then in 1990, a group published the first evidence that it was the shortening, the gradual shortening of telomeres that correlated with aging in a test tube or in vitro. Now, of course, there's a lot of correlations in science, but it certainly didn't prove cause and effect, and maybe this was only something in tissue culture which may not be replicated all the way into human beings. So at the same time, another group actually started measuring telomere lengths in real human tissues. And as you can see on this slide, that if you look at fetal or reproductive tissues, that telomeres in humans are somewhere between 15 and 20 kilobase pairs at the ends of our telomeres. And as we get older, in all of our cells that divide, you see gradually shortening of telomeres. And it increases basically throughout life until when you're in your 70s or 80s, most individuals have telomeres in the very critically short amounts. And so how does one think about why we would limit the number of divisions that human cells should be able to undergo? And the idea that has been proposed by our group and others is that aging at the cellular level may be, in fact, a potent anti-cancer protection mechanism. That if you imagine it takes many alterations in one cell for a tumor to actually emerge, one can imagine if you limited the maximum number of divisions that a cell can undergo, that in fact you could reach a, quote, brick wall before you got that last hit that would allow your cell to become a cancer cell. So maybe in long-lived species such as humans, telomere shortening actually evolved as a mechanism to protect us long enough to reproduce and nurture our young in the absence of getting dreaded diseases such as cancer. So everybody knows that aging in cancer is basically a double-edged sword. It turns out that approximately 70% of all cancers occur in the 65 and older segment of the population, and we need to know more about why this occurs. It turns out that if you look at telomeres in patients with diseases that are not quite cancer, we call these pre-malignant diseases, often they're called carcinoma in situ, and here's just a long list of breast cancer, ductal carcinoma in situ. That's the lesion that occurs just before you get real breast cancer. And prostatic intraepithelial neoplasia, or PIN lesions. That's the cancer type that the pathologists see that make you at risk for prostate cancer, but you don't have prostate cancer. Well, one can look at those tissues and measure their telomeres. And here is the method by which this is done. It's called fluorescence in situ hybridization, or FISH. and it's used for, specifically in this instance, for measuring telomeres. So FISH provides a sensitive method for measuring these, and I'm not going to go through the details, but to say you can actually make a probe that will hybridize very specifically, very uniquely to the telomeres and only to the telomeres, and using quantitative biology approaches can accurately determine how long the telomeres are. And you can do this with a variety of ways. And if you look at a tissue like from a patient with ductal carcinoma in situ, you would see cells that look something like this. You would see interface cells on the bottom left of your screen, and you can see what I've done here is I've labeled these with two different colors, pink being the centromeres of each chromosome, and green in this case, the telomeres. And in the interface cells, they're all mixed together. And in the middle, you see the metaphase chromosomes illustrating the telomeres at the tips of all of our chromosomes. Now, if we were to take a tissue like we have here, I think you can see where I have the arrows showing luminal epithelial cells, there's almost no red or telomere signal, whereas the cells around the luminal epithelial cells have robust telomeres. So the question that arose is this growth arrest that these luminal epithelial cells are undergoing a mechanism to protect those cells from making that next step and becoming a cancer cell, or is it in fact the initiating event in genomic instability that eventually leads to the cancer cell? In order to deal with that, one has to do more experiments. So what I've said so far is most premalignant tissues have very short telomeres, and that telomere attrition may not only be a tumor suppressor pathway, but in combination with perhaps some of these steps that are important leading to the initiation of cancer, they actually help drive the genomic instability that we know occurs in most cancers. Now, alternatively, in order for a cell to bypass this senescence growth arrest, the cell has to figure out a way to deal with the telomeres. If the telomeres can't get any shorter, the cell will simply not grow. And one of the hallmarks of cancer is that the cells are almost always capable of indefinite proliferation. They're basically, essentially immortal. So in order for a premalignant cancer cell to become a cancer, they must engage a mechanism for stabilizing the telomeres for the advanced growth of the cancer. And the answer to that, of course, is telomerase. And a number of years ago, I actually spoke at this conference when we were first starting to discover things about telomerase. But it turns out now we know that telomerase is detected in almost 90% of all human cancers. We have very sensitive assays for not only measuring telomeres, but also for measuring telomerase. And what telomerase essentially does, it's a cellular reverse transcriptase. It basically goes to the ends of the telomeres, and in every time a telomere shortens because of the end replication problem, telomerase adds back telomeres. It's a very amazing system, and yet it works very efficiently, and as I said, it occurs in almost all human cancers. Those cancers that do not have telomerase often result in very good outcomes for the patient. So it is possible to have a cancer that has not engaged this pathway, and those cancers almost universally go into remission, especially pediatric cancers. It turns out the discovery of telomerase and how it works was awarded not only the Lasker Award in 2006, but was awarded the Nobel Prize in 2009 to Elizabeth Blackburn and her colleagues Jack Shostat and her student Carol Greider. So, what is the evidence that telomerase has anything to do with things? Well, it turns out in experiments we did over a decade ago, we showed that introduction to telomerase into normal cells that would normally senesce, in fact, elongated their telomeres, as you can see in the middle panel. And as you can see, instead of undergoing replicative senescence, that's the right panel, the cells continued to grow indefinitely. So I just told you telomerase was on in 90% of cancer cells, and now I'm telling you I can take a normal cell and put telomerase in. So the question you should be asking yourself is, are these cells that we put telomerase in cancer cells, or are they normal cells? So we addressed that question, and what we found is that immortalization of normal cells with telomerase does not by itself transform cells. In fact, the cells have all the hallmarks of normal cells, and in fact, the only difference is that they can grow indefinitely, and that turns out to be a hallmark of some stem cells in our body. So we're at this crossroad, right? Come on, it's one or the other, aging or cancer. So telomere biology and telomerase are at this bridge between what we know at the mechanistic level about cancer and about aging. And what we need to do now is to pick how we go about dealing with this. So on one hand, telomerase expression in cancer cells doesn't drive the cancer process, but is in fact permissive for cancer development. And therefore, one could argue, perhaps, if you could inhibit telomerase in cancer cells, this might be a potent cancer therapeutic approach. On the other hand, as long as you put telomerase in normal cells without other alterations on the way to becoming cancer, this could actually be very important in what we all call rejuvenation or rejuvenative medicine. And I will talk briefly about both these. I don't have time in this short overview to talk about all the clinical trials that are currently ongoing. This is a quick slide to show you that there are a number. There's at least 3 major clinical trials, some in phase 3, some in phase 2, that are currently ongoing with active inhibitors to see, in fact, if we can impact on the cancer process by inhibiting this enzyme. Now, what is the evidence for telomere shortening in human genetic and chronic diseases? Because this is where rejuvenative medicine actually becomes more important. And here is a long list of things that I don't have time to cover all of them, but chronic wounds, many of you know about, pressure ulcers, diabetic ulcers, often have critically short telomeres. People with myeloproliferative disorders have short telomeres. There's a lot. The leading cause of age-associated blindness, macular degeneration, the retinal pigmented epithelial cells have short telomeres. So the list goes on and on. And it turns out in the last 5 years, we've discovered genetic diseases, which we now call the telomeropathies, that are due to changes, genetic changes in the genes that regulate our telomeres and this telomerase enzyme. And what I'd like to do is just show you a couple of these. One is called dyskeratosis congenita. These patients usually present very early in life with abnormal skin pigmentation. They have cutaneous problems, nail dystrophy, but most obviously, and what they usually go to the doctor about, is they have anemia. They basically are in bone marrow failure. When you look at the bone marrow of these patients over here on the left side of the slide, you can see these formed elements in the bone, but all these white large areas are really fat cells. This is a 19-year-old patient that's bone marrow should be absolutely full of hematopoietic precursor cells, but instead these people have essentially lost most of their stem cells in their bone marrow. And most of it is due to having mutations in genes that regulate telomerase and telomere length. These patients, about 20% of them, present with pulmonary disease. These are people that have what we call pulmonary fibrosis, as shown in the pictures, the histology pictures, as opposed to normal lung in the top left. People with pulmonary fibrosis replace their normal air sacs that you breathe and exchange air with fibrotic tissue, and this actually leads to great problems and affects 90,000 people in the United States each year, and a third of them die each year. And it turns out if you look at the telomeres in these patients with pulmonary fibrosis, it used to be called idiopathic pulmonary fibrosis because we didn't know the origin. But now we've actually sequenced the genes, and you can see here that patients with the disease generally have shorter telomeres compared to age-matched controls. This curve going down is, in fact, the patients, normal individuals, showing the gradual loss of telomeres throughout age. Every patient, whether they've got the mutation with the disease or without the disease, fall below the line, suggesting that these people are born basically with half the amount of telomere maintenance as a normal individual. So the idea is that these telomeropathies result in premature telomere shortening, cell cycle arrest, probably loss of progenitor stem cells, and that leads to clinical disease. So it suggests that just losing half of your telomerase may lead to this stem cell depletion, and it argues that telomerase is not in excess in the body, and perhaps if we could come up with methods to slightly augment telomerase in these patients, we might have a wonderful therapy for replacing their bone marrow cells either in vivo or ex vivo and then giving them back their own cells. So this is sort of a cartoon to illustrate sort of what we think. As we get older, telomeres gradually shorten, and if you wait too long, you wind up having other changes leading to cancer. If we can intervene in some of these younger individuals with telomeropathies, it's possible that we might be able to transiently activate telomerase and give these individuals the telomeres they need to sustain their bone marrow so they won't get sick. So for every good thing, there's a bad thing. So obviously, putting telomerase into normal cells would slow the rate of telomere loss. It would improve some of the extracellular matrix and immune cell structure and function. It would prevent or slow down the rate of genomic instability, which might reduce the incidence of cancer. It activates renewal pathways. That's been shown by many publications. And it also may increase repair and resistant to stress-induced apoptosis. However, introducing telomerase uncontrolled could, in fact, increase the change for additional mutations to occur. Cancer cells have abrogated this pathway by upregulating telomerase, and the worrisome thing is that increasing the doublings too much may result in an increased risk of mutations leading on to cancer. So one would have to be very careful in the application of adding too much telomerase to cells so that we don't increase the risk of developing cancer. So everybody's interested in aging. All of you that are here, at least I hope, are interested in aging. And centenarians are sort of the tip of the iceberg. These are the individuals that live to 100 years or longer. And we'd all like to know, what's the secret to living to 100 years? Well, it turns out in the 1950s, only a few thousand centenarians were reported worldwide. In 2009, over 300,000 individuals have been recorded with accurate records of being over 100 years old. And it's estimated in the next 30 or so years that this is going to increase dramatically. Improvements in biomedical care and intervention in diseases such as stroke and heart disease and hopefully cancer in the future is going to result in aging-associated problems being the key thing that is going to limit maximal lifespan. So we wanted to ask the question, what's the relationship between telomeres and aging in real people such as centenarians? So you can do a number of studies, and because the time is short, I won't go through this, but it's called SNP analysis, single nucleotide polymorphism discoveries, to see if we can find if there's something in the genes of individuals that are centenarians that makes them live as long and as healthily as they do. So I won't go through the details of this, and I'm going to actually skip this slide with the real data and just come to the summary, which is to say, based on significant amount of research we did with the people in New York at Albert Einstein College of Medicine, we published last year that a significant number of centenarians have telomeres similar in length to much younger individuals, often those 60 to 70 years old. So essentially, these people have 30 or so years of extra telomere length compared to age-matched controls. Now, many centenarians are just lucky, but about 20 to 25% of this population had longer than expected telomere length. It also suggests that telomere length is inherited. We always knew that if your parents lived a long time, the odds are you would live a long time as well. This has also been documented in mouse models of aging, and this is work, again, I won't go through the details, from Ron DePinto, the president of MD Anderson's lab, where he showed if you knocked out telomerase, even in mouse models, mice will undergo premature aging. They get alopecia, that's premature graying of the hair. They don't heal wounds as well, and they have fertility problems. So there's evidence not only in humans, but also in animal models that telomere biology is actually important in the aging process. So the question that emerges now is our telomere test, ready for prime time? Just this April, Science magazine, the most famous of all the sort of publications, came out and did a large study on all the different commercial and different types of research-based telomere tests and asked the question, are we really at the point now where we should be applying telomere testing to the vast majority of the population? So the answer is, we know very little about the dynamics and telomere lengths over multiple years and large cohorts of human populations. Most of this work has been done in basic science laboratories such as my own. And it's unlikely in these economic times that government agencies are going to fund the tens of thousands, if not hundreds of thousands of studies and follow patients year after year to really validate what is the importance of telomere research and aging. Therefore, the private sector is going to have to step up and determine if all these associations that have been reported in the literature of diseases with telomeres and length of telomeres hold up in well-controlled experiments. So why should you care about how long your telomeres are? Well, this is a busy slide, but let me just go through it quickly. Commercial telomere length tests are now a validated biomarker of aging. This is very similar to when cholesterol tests first came out. We didn't have Lipitor when we first started measuring cholesterol, yet now millions of people get their cholesterol test done, they get your blood pressure test done all the time, and it's my expectation in the future telomere length testing will be just as popular as cholesterol testing. And while right now there are no FDA-approved prescription drugs to reverse telomere loss, there is substantial evidence that certain lifestyle habits can modify telomere length and influence the aging process. And there's also, from some of the people here at this meeting, emerging classes of natural products such as telomerase activators that may influence aging, and we need rigorous scientific biomarkers to prove the mechanism of action. So it's important that doctors and patients start these longitudinal telomere measurements as soon as possible, including getting complete family histories, because by knowing how long your parents lived, what your BMI is, what your cholesterol level is, whether you smoke or not, how much you drink, all of these things can potentially influence telomere length maintenance, and hopefully some of these lifestyle modifications could reverse some of these telomere losses that we know occur under certain conditions. So these are the tests that are out there commercially right now. Some of these companies are here, and you can visit them at their booths. There is the very popular qPCR technique, which is over here on the left, which is good because you can do it with multiple samples, and it's susceptible, obviously, to high throughput. There is the Lansdorf model of flow cytometry, which means you take a blood sample and you run it through a specialized piece of equipment, and that also is pretty good. It uses the same technique as FISH, as I described earlier. You label up the telomeres, but now instead of looking at it in a microscope, you use a fluorescent activated cell sorter to look at it. On the right is the Blasco life length technology, which is high throughput quantitative FISH analysis. More to say about that. And finally, at the bottom, there's a research-only technique, which is called STELLA for single telomere length analysis, which is very cumbersome, takes a week to run an assay. So that's not ready for prime time. But QFISH, telomapping, I've already showed you one image, and I'll show you one image of that as well. So how do we do high-throughput telomere length measurements? One uses multi-well plates, like 96 well plates. You plate these out, you incubate them, and as I showed you before, you hybridize these cells to a telomere PNA probe, and then you run it into this very expensive instrument, which will quantitatively measure the length of the telomeres and tell you everything you want to know about every one of your 92 ends and every one of your cells. And one can measure two things. One can measure average telomere length as seen on the end, on the left, or you can look at the percent of short telomeres. Only QFISH on tissues and QFISH on blood samples can actually provide detailed information about the percent of short telomeres. And it's now known in a variety of diseases, it's the short telomeres that limit the growth of cells, not the average length of telomeres. And here's a study that was published just this year from a Spanish group showing that in certain inherited types of chromosome diseases, such as breast cancer, the ones in black are the shortest telomeres, and that correlates with the incidence of cancer in these patients. So it's the short telomeres that count. Telomapping, or tissue telomere FISH, is shown here on a normal prostate on the left and a prostatic interepithelial neoplasia on the right. As you can see on the right, it's the luminal epithelial cells where there's no red dots. that means those cells are undergoing growth arrest. And I ask yourself, if a doctor was to take a biopsy of this tissue with normal tissue and with this pin lesion, and you just took the average telomeres, would that be very useful? And the answer is probably not. You need to know where the cells with short telomeres are and how many they are and how short they are. So it's the shortest telomeres that lead to age-associated diseases. This is just to show you the same thing, that when a human cell gets old, it gives off a DNA damage signal, which we can measure in cells, and that always correlates with the shortest telomere. So I'm just coming to the end. What are the issues that one would want to consider when deciding to test for telomere length? First of all, what is the reliability of the assay? Is the test accurate to within 5%? If one conducted the same experiment with 3 samples from the same patient over different times, that's important to know if we're going to make advances with longitudinal studies. Does the test provide both average and percent of shortest telomeres? I've already talked about that. Average telomere length may be less useful since a single critically short telomere may be sufficient to initiate cell senescence. And finally, it's important to have detailed questionnaire and family histories so that one can actually, in the future, be able to correlate all of these tests so we can make more meaningful insights. So in conclusion, what I've tried to tell you in this brief overview is the analysis of human telomere length has become a central validated biomarker that reflects biological, not chronological human aging processes. It's really important to distinguish those two. Accurate commercial tests are now becoming available that will lead to better understanding of the relationship of telomeres and telomerase to aging and cancer. Products currently being touted to promote healthy aging may wish to consider incorporating telomere length measurements or testing to determine if telomere biology may be the mechanism responsible. And I would like to just thank Maria Blasco, the director of the CNIO in Madrid, Spain, also one of the founding scientists of LifeLength, and Steve Maitland, the CEO of LifeLength, who is exhibiting here if you want more information about the various tests. And I think there's another company here, also SpectraCell, that's exhibiting here. So please go by and see them, and TA65, who has a telomerase activator, so you could find out more about this new technology. And on that point, I'll stop, and thank you for your attention.